Hello boys, welcome back to another session of video tutorials, and this session is meant for class 11th biology students, and this is meant for 26th July 2020, and our memory helps us in tracing out what we have learned in our previous session. We were studying about structure of proteins, amino acids, and we were also discussing and reflecting on structure of enzymes, classification of enzymes, and what are the various functions that these enzymes perform. And in today's session, we are going to discuss and reflect upon the various types of enzymes or groups of enzymes and enzymatic action, how exactly an enzyme takes the action. And we were also discussing factors affecting enzyme activity and then inhibition of enzyme action. These are the topics that we will discuss in this session. To start with, as all of you know, enzymes are organic catalysts that catalyze biochemical reactions without being utilized themselves. And I compared the enzymes with cricket umpires, if you remember. Like cricket umpires, they do not play a direct role, but they ensure that rate of reaction goes on and the chemical reactions take place in an ordinarily fashion. And in this session, as I said, broadly enzymes are classified into 6. The first one is oxidoreductases. These catalyze oxidation as well as reduction between two substrates, that is S and S dash. That means transfer of oxygen and hydrogen atoms or electrons from one substrate to another. Example, dehydrogenase oxidase. And the second one is transferases. These catalyze transfer of a group, that is other than hydrogen, between a pair of substrate S and S dash. So they are responsible for transfer of specific group from one substrate to another. Example, transaminase. And then third, hydrolases. They catalyze hydrolysis of ester, ether, peptide, glycosidic, carbon-carbon chain, carbon halide chain, and the phosphorus and nitrogen bonds. So the hydrolysis of the substrate is done by these hydrolases. Many digestive enzymes are the examples of hydrolases. And fourth group is isomerases. These catalyze interconversion of optical, geometrical, and positional isomers. That's why the name isomerase. So these are responsible for change of molecular form of a substrate. Example, phosphohexoisomerase. And then fifth one, lyase. Catalyze removal of groups from substrates by mechanisms other than hydrolysis, leaving double bond. So non-hydrolytic removal or addition of a group to a substrate. Example, decarboxylase aldolase. And sixth, ligases. So ligases catalyze the linking together of two compounds. Example, enzymes which catalyze joining of carbon and oxygen, carbon sulfur, carbon nitrogen, phosphorus oxygen bonds. As we said, ligase means something that joins. So joining of two molecules by formation of new bonds. Example, citric acid synthetase. And we talk about the enzymatic action, and before that we discuss how exactly the complex is formed. Enzyme action starts. As you are seeing, this yellow color one is an enzyme, and it has got something like key and lock mechanism, I said, or lock and key mechanism. And the substrate is in green color. So the substrate also has got some grooves, so as enzymes. So where the grooves are there, that's called active site in an enzyme. So the number of substrate molecules converted to products. Look at this. In the first one it is substrate, and the last diagram it is products. So the number of substrate molecules converted to products per minute by an enzyme is called turnover number, and this exhibits enzymatic efficiency or efficiency of an enzyme. So all enzymes have a specific 3D structure, and a part of it is known as active site, into which the substrate having some grooves fits in. The point where substrate is bound on the active site, you can see in the second diagram, is known as substrate binding site. There is an obligatory formation of E and S. E for enzyme, S for substrate. So in the second figure you are seeing enzyme substrate complex being formed. We say ES complex. This complex formation is transient phenomena. It keeps on changing. During the state where substrate is bound to the enzyme active site, a new structure of the substrate is called transition state. Structure is formed. Very soon the product is released from the active site, as you are seeing in the third figure. If P is at lower level than S, that is substrate, P for product, the reaction is an exothermic reaction and is not supplied energy by heating. So you can see here the graphical representation. In order to form a product, however, whether it is exothermic or spontaneous reaction or an endothermic, energy requiring reaction. Here you can see the activation energy, and we can focus on three things. That is higher state of energy or higher energy state or transition state. Now you can see first one graph, that is on x-axis you have got a reaction progress, and on y-axis you have got energy, that is potential energy P, and in the central you have got a dotted line, that is initial energy content of the substrate, that is the activation energy line. So the difference you can see, first we have got one graph which says energy release as product forms and activation energy without enzyme top and activation energy with enzyme. Now see the difference. Activation energy without enzyme is very large. Activation energy with the presence of enzyme is very narrow. And see how quickly the product is formed. If there is an enzyme present in the reaction, the substrate S is getting very quickly converted into P products. So the products are formed very fast with optimum levels of energy, with the minimum levels of energy. If there is no enzyme, look at that, the graph is going up, and that's how activation energy is required in huge quantity, and the products that are formed also will be very slow. So the difference in average energy content of S from that of this transition state is called activation energy. Enzymes eventually brings down this energy barrier. As you are seeing, the energy levels are being brought down by the enzymes, and there are two viewpoints by which enzymes are sponsored to by which enzymes are supposed to bring about chemical reaction, and that is lock and key mechanism that I explained to you. So what affects this graph? There are two things that will affect this graph. One is the temperature. If the temperature is optimum level, then the something like optimum temperature for enzyme activity is let's say around 30 to 40 degrees centigrade in animals and 20 degrees to 30 degrees in plants. Then this graph will be very fast, quick, and the final products will be formed fast. On the other hand, if the temperatures soar up beyond 40, say 45, 50, then denaturation of enzymes takes place. As a result of denaturation of enzymes, the products will not be formed. The graph gets affected. Similarly, the low temperatures, sub-zero temperatures also, enzymes get denatured. Naturally, products cannot be formed. So, temperature plays a crucial role in making the enzymes active or inactive. And the second factor that will affect the enzymatic activity is substrate concentration. How much substrate concentration we have taken. And if you plot the graph, there is the two scientists, we must be thankful to them, that is Michaelis and Menten. So we must remember this Michaelis and Menten equation. To determine the effect of substrate concentration in enzymatic reaction, Leonard Michaelis and Maud Menten in 1913 proposed a mathematical model and derived a relationship which is mathematically expressed as V is equal to Vmax S divided by Km plus S. And in this equation, it's there in your textbook. If you see that the Km is Michaelis-Menten constant, and the substrate concentration to produce half maximum velocity. Graph also is there in your textbook. You can keep the textbook by the side of you and you can see it. And then V is velocity of the reaction, Vmax is maximum velocity, and of course S is substrate concentration. So with increase in substrate concentration, the velocity of the enzymatic reaction rises at first. To begin with, it rises. The reaction ultimately reaches a maximum velocity, that's called Vmax, which is not exceeded by any further rise in concentration of the substrate. This is because the enzyme molecules are very few than the substrate molecules, and after saturation of these molecules, there are no free enzyme molecules to bind with the additional site. That is the reason why the concentration, the speed, the maximum Vmax will reach to certain level. After that, beyond that, it will not go. And then we can see here the substrate enzyme being formed, and then we have got what is called some substances will form, some substances will duplicate, will appear as though they are like original substrates, but they act reverse to substrate. They are called inhibitors. So the activity of enzyme is also sensitive to the presence of specific chemicals that bind to the enzyme. When the binding of the chemical shuts off energy activity, the process is called inhibition, and the chemical substance that inhibits is inhibitor. You can see here inhibitor. In the first reaction, you can see enzyme is there and the substrate, lock and key mechanism. So to the active site, the substrate is fitting in. You get that enzyme substrate complex. After that, because of the substrate undergoing chemical reactions, you are getting products. This is normal way of enzymes helping the chemical reactions to undergo and form products. Whereas there are certain substances which will act as inhibitors. Now look at that, enzyme has got a particular groove and the inhibitor is also fitting in. So by fitting in, it will not allow the enzyme to do what it is supposed to do. As a result, inhibitor competes with the substrate and blocks the chemical reaction. So based on that, we have got reversible inhibition. This is temporary, of course, and it can overcome by increased concentration of substrate, dilution and dialysis. So you can use three: increase or decrease the substrate concentration, dilute the substrate concentration, and dialysis. So we can overcome the reversible inhibition. What about irreversible inhibition? It's a permanent nature, in which the inhibitor combines with a specific functional group of enzyme through covalent bond. On the basis of competitiveness, enzyme inhibition can be divided into competitive inhibition, non-competitive inhibition, and uncompetitive inhibition. And the competitive inhibition is reversible, occurs due to substrate or increase enzyme analog. Non-competitive inhibitor forms a complex with enzyme at a site other than active site. You are seeing active site, other than active site, the competitor goes and sits and thereby changes the structure. Allosteric inhibition, inhibitor binds to enzyme at specific site other than active site and change the structure of active site so that it affects the substrate binding. That's how it is allosteric inhibition. And there is a feedback inhibition also, an enzyme regulatory mechanism where a product or intermediate of reaction functions as a temporary inhibitor. This combines with the regulatory site and its concentration crosses the threshold value. The end product inhibitor functions as a negative modulator, and the enzyme inactivated is called allosteric enzyme. So Michaelis-Menten or Km constant is not applicable to allosteric enzymes. So this is how the enzymatic reactions will be having their own controls, and the main problem in this is the inhibitors. So with this we are completing the chapter biomolecules, and what is that I want you to do? First, draw the graph. Two graphs are there in your textbook. Draw the graph. One is the process of reaction, enzymatic reaction, the one that I have shown you here, and try to understand how this graph can be inferred. And the second graph is there in your textbook. Please go through that. Effect of change in concentration of substrate on enzyme activity, Vmax. So I have given an explanation now. Please draw the graph, understand what is Vmax and what is Km.